position
So
of the
here.
eye.
Yes, just drop the mic.
Okay, so what I'm going to do is I'm going to narrate it myself because...
You can put the caption so it makes it easier for you.
Ah, yes, caption.
There we go.
Okay, so anyway, this image actually perfect for your lab because this is what we're going to look at actually, how the skeletal muscle, how the cardiac muscle, and the smooth muscle looks like in the microscope. In terms of how they really work, we're going to do it in the cardiovascular and the GI. But what I wanted to show you guys was in this video, not really this part, because that's already actually in the lecture. It's more like that the entire muscle is built up from elongated structures, one by one, from larger to smaller, one by one. There is a kind of like a hierarchy. You will see right now as he cuts the biceps brachii muscle. So that's your first sausage, that's what I call it, like this elongated structure. You see, he cuts the biceps, and now we can see what's inside. So this was sausage number 1. Now let's look at the internal. So what's now is talking about the connective tissue covers, which is identical, guys, with the nerve. You see, the purple one is the epimysium instead of epineurium. Then here's a fascicle. The green you see everywhere is going to be the perimysium instead of perineurium. And now, pulled out one muscle cell, skeletal muscle cell, this is where you can get lost. So this is actually one fascicle. One skeletal muscle cell is pulled out and now covered with, what comes next? Endomysium, right? Instead of endoneuron, you're gonna say endomysium. So again, the same idea, collagen covers, supporting the muscle, protects it from tearing up, blah, 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 blah. More functions are in the PowerPoint. But what is really important to see that, you see here's a muscle, or you see a biceps like this, right? So the epi, peri, and endomysium actually comes out at the end of the muscle, kind of merge with each other, and that's your tendon. But we didn't have that in the neuron, right? Now, you see, now we are going intracellularly, internal structure of skeletal muscle cell. You see, next sausage is coming up. The cell itself is as long. How long it is, guys? As long as the muscle. So if the biceps, I don't know, 30 centimeters. Sartorius muscle, you guys are gonna learn, 1 meter long. So, of course, in the eye it's smaller, obviously, but the big skeletal muscles are large, and the cells are as long as the muscle itself. So, you see, now they open the plasma membrane that we're going to call sarcolemma, so you can see the organelles sitting in the cytoplasma that we are going to call sarcoplasma. Okay? And what we're going to analyze one by one is the T-tubule, the sarcoplasmic reticulum, the terminal cisternae, and mitochondria. So lots of structures inside the cell. You can hardly see any cytoplasma, guys, because look at it's all filled out with these purple pink fellows. That's going to be your next sausage. Let me see this guy shut up and moves to the here we go. You see? The entire cytoplasma or sarcoplasma is filled with myofibrils. So you can hardly see actually cytoplasma here. It's all squeezed, okay? Because you have these myofibril organelles, and it can be between 100 to 1,000 little sausages nicely lining up inside the cell. okay? This is where it's, you know, you have to follow me. So how many sausage did we see so far? Okay, so we did the muscle itself. Then we did the fascicle. Then we did the skeletal muscle cell. And now we are going inside, intracellularly, so cell biology, okay, is filled with myofibril. So myofibril is already an organelle. Okay, so how is this myofibril? And again, myofibril is again as long as the muscle cell itself. So if let's say, as I told you, the muscle cell was 1 meter long, then the myofibril is also 1 meter long. Okay? So now they magnified one myofibril from one muscle cell. And again, I suggest don't use the word muscle fiber because you're gonna get lost in the sausages, okay, honestly. Just better to spell it out, skeletal muscle cell. And that's when you're gonna start seeing the sarcomeres. This is one sarcomere. So what is a myofibril, this long structure? Millions of sarcomeres in a chain connected with each other. Sarcomere. And sarcomere is considered the structural functional unit of the muscle. Okay? So the myofibril is an organelle made out of, only God knows, let's call it 1 billion, sarcomeres next to each other. Of course, it depends on how big is the muscle. Because let's do it again. The muscle, the size of the muscle, the size of the skeletal muscle cell, and the size of the myofibril inside is the same. Okay? And now comes the little pieces of sarcomeres, which is basically as a chain connected with each other makes up the myofibril. Okay, so let's move on. What did I do? Oh no, I need help. Go back. No Grammarly, okay? Bye-bye. Bye-bye, lady. How do I go back, guys? Somebody help me? This one? Maybe I did it. Thank you so much. Okay. Yeah, it is. Not now, okay? Not now. Okay, so here we go. Let me just, so I push this one, right? Okay. Don't go away in case I need you. I shouldn't have stopped it. So where is the CC now? Okay, so now it's, thank you so much. I think it's, I'm not gonna stop it again.
I'll wait here.
Thank you so much. But anyway, now comes the the structure of the sarcomere. And you need to understand that this is an electron microscopic image of a sarcomere. And when they looked at this image, that's what they drew here on the bottom. So one sarcomere goes actually from Z line to Z line, this black line. On the image, Z line to Z line. I don't know why the CC disappeared. Okay, here we go. Okay, and then what you're gonna see is white, black, white, black, white, black, white, black. So what did I just describe to you guys? The striations. So how do I see the striations of the skeletal muscle? Usually I just say it looks like a zebra, right? Like a zebra, it has black and white lines. Because if you imagine 1,000 myofibrils parallelly lined with each other, everything is lining up. The dark bands and the light bands, and the dark bands and the light bands. So eventually you're gonna see a striation as it gets bigger, and you can see it in light microscope and you can see it in electron microscope. So what is the dark band, guys? The A band. You see the A band? That's where the thick myosin filaments are sitting. And the white one is the I band, This is the summary, guys. See, these are the sausages. You see that? We started with the muscle, fascicle, muscle cell, myofibril, and now we are here at the myofilaments. Okay? And I think after this I'm going to stop the movie, but I think this is a very good summary to see the layers of organization. Every structure is getting smaller and smaller and so on. So you are going from macroscopic level to microscopic level, including electron microscopic level. Or you could say you go from organ level, which is more like macro, to all the way down to microscopic level and even chemical level. Okay? So this is what I was explaining to you guys a minute ago. What is it? Okay, anyway, I don't think we need this anymore, thank you. So this is the way a muscle is organized. So I do recommend that you guys watch this again, okay? And I would like to continue to show you quickly. Thank you so much. Okay, so here's your YouTube video when you want to look at it. Just a couple of things, just boom, 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 okay? I put here for you a little terminology. Make sure that you look at it. Myo means sarco, which means flesh. Whenever you see these prefixes means the beginning of the word, it's muscle, okay? And you're gonna use them all the time. Objectives, and then these are the functional characteristic of muscle. I leave that to the physiology people, okay? But it's all, basically, you see I underlined for you the keywords. Receive, respond, shortens, can be stretched, they can recoil. Okay, these are all the characteristics. And, guys, I'm going here a little faster because this is the easy part. This is nothing difficult. Now, the classification of muscle tissue based on structure and function, okay? Structurally, either zebra or no zebra. Striation or no striation, okay? And striated can be the skeletal or cardiac muscle. The no zebra is the smooth. So because there is no striation, no zebra, we call it smooth. And now comes the physiology part. If they are voluntary muscle, they are the skeletal. If they are cardiac, they are involuntary. And smooth muscle is also involuntary. So now you the whole thing put together, you can say that the skeletal muscle is striated voluntary. And this is what we think about skeletal muscle. All on the bones, helps you walking, and keeps your posture, stabilize joints, and very important, generate heat. Okay? So skeletal muscle is, people usually know quite a bit about skeletal muscle, right? Now comes the cardiac muscle. Now this is striated but involuntary. There's only one place, guys, in the middle layer of the heart that we're gonna call myocardium or myocardium. What is this guy doing all day long? Pumping the blood. Halas, okay? Now let's look at the smooth muscle. This is what you're gonna call non-striated involuntary muscle. Okay? And where is it going to be located? In the middle layer of all hollow organs. And I showed you a couple of hollow organs, which means those organs that have a lumen in the middle. And in the lumen there is some stuff sitting. You remember we did the kidney and the pee is sitting, and we did the esophagus and the food is going, and the duodenum and the poop is going, right? So we are dealing with all these organs that has a lumen. So I put it here for the muscular tubes with lumen. So what is really the smooth muscle is doing in the middle layer? When it contracts, it's gonna push forward the stuff in the lumen. And I put here for you, very important, everything is in one direction. Okay? Physiologically healthy individual, everything is moving forward. Because if some stuff is moving backwards, that's already a disease. Just think about when you are vomiting. Food is coming back from the stomach. Is that a good thing? How about vomiting poop? Do you know that that can happen? unfortunate, in an extreme case, is that a good thing? So everything has to go forward. So the two major location is really blood vessels, okay, and again in the middle layer. And what is it going to do? Push the blood forward, and also will contribute to the blood pressure. And the digestive organs, the big word you are looking for is peristalsis, okay? And finally, I wanted to ask you, because you should be able to know, if the middle layer is the muscle, what is the inner layer? What? Somebody said something? Because that's what we covered. The epithelium comes first. You remember? That was one of the definitions. Epithelium is looking at the lumen. Okay? So now you already know the structure of hollow organs, actually. You need the epithelium, you need the smooth muscle, and then comes the little connective tissue, connect the structure to the rest of the body. Done. Okay, so now that we got this straight, let's move to the location of other striated voluntary muscles. So the ones that are attached to the skeleton moves the body, blah, blah, blah. Then there are visceral striated skeletal muscle. All you need to know is viscera means organ. So in the soft organs, some of them has skeletal muscle, okay? And they are not attached to bone. The tongue, the pharynx, the esophagus, and the diaphragm. Okay, and finally the facial muscles. Skeletal muscles that we are making smiling and crying and all of that stuff is inserted in the skin. Okay, so on the next slide talks about muscle-like cells, okay? Just in general, you already know the myofibroblast. We did that before. It develops from fibroblast and works through repair and makes scar tissue, which is basically collagen. But again, myo refers that it's a little bit like muscle because it has contractile proteins, actin and myosin. It's fibroblast because it's like a fibroblast making collagen. So it's both at the same time. Pericyte is going to be more the objective of the cardiovascular system. When the capillaries when you hit the capillary levels, the thinnest blood vessels, they are very lonely, actually. They don't have tunica, they don't have smooth muscle anymore over there, what we just did. Blood vessels, smooth muscle. Capillaries has nothing. So the best friend he got is the pericyte. And what is the pericyte is doing? What the glia cells are doing. Kissy, kissy, huggy, huggy of the pericyte. So people think that this is blah, blah, blah control. Lots of theories, probably it's the stem cell, and it's also a little bit contractile, which means that it kind of replaces the function of the smooth muscle that is missing. Okay, and you have myoepithelial cells, that there's an image for that. I hope you recognize the flowers of a gland. And the ocini I covered with myoepithelial cells. So when these cells are contracting, then the secretion is pushed from the ocini. Oh, wait a minute, I forgot to turn on this thing. Sorry about that, guys. Somebody, too many drama this morning, so. Okay, so they will help to push the secretion from the ocini into the duct. Okay, now comes the histology, and this is what we just did, okay, in the movie. So I'm gonna skip over a couple of parts, but again, do you see what here is the 7 sausages, so to speak, right? And we went up till this point, that we looked at the sarcomere. And we didn't look at the myofilaments yet. So I may just jump over there. But you see, everything is elongated structure. 1 to 7. 1, 2, 3, 4, 5, 6. Hey, where is the 7? Yeah, the 7 is basically is here, but they don't show you the individual molecule. The individual molecule is in the filament, which is number 6. One more thing, did you see that I underlined fibrous filaments? We did talk about this briefly in last week when we talked about collagen, that Which one is the biggest fiber? The fiber. Then when it gets smaller, you're gonna call it fibril. When it's like really thin, it's filament. So the name already tells you the size. Okay? This is kind of the beauty of Latin terminology. And again, what I usually say is if the word gets longer, the structure gets smaller. You see, the filament is the smallest, and that's the longest word. Okay, so here's the organ level. We did that with the movie. Okay, here is the epiperi, endomysium, blah, blah, blah. Same thing. The only thing on this slide for you guys is that obviously, which one is the thickest collagen? The epi, the peri, or the endo? Of course, the outside one. Okay? And the thinnest in the middle is actually type 3 reticulin. Okay? Yeah, one more thing I want to emphasize for you guys that most books don't say it, so I had to dig it out from all kinds of, okay, that one fascicle is, let's say, 150 skeletal muscle cells in a perimysium. Okay? Now this one summarizes for you the functions of the connective tissues. Whatever collagen can do, it's happening, right? But I told you making tendons, but the other ones are maybe more new for you. Transmit the force. We are going to create a force inside the muscle by the actin and myosin action. And that force actually had to be transmitted eventually to the bone, because the bone is moving in the joint. So transmitting force. Then another one is that you can see very nicely on this image that this is the highway for blood vessels and nerves. Obviously, where are going to be the bigger one? In the epimysium. Then they're going to start branching, getting smaller, and entering the perimysium. Branching, getting smaller, and they're going to enter the endomysium, so that endomysium covers every single cell, so that every single cell is going to get oxygen and nutrient. However, this is what I really want to emphasize for you guys, that, and I didn't know where to put it, so I put it here. Muscles, guys, is different from other tissues. They need not only an oxygen and nutrients from the blood to survive and be happy, they also need action potential. If the nerve gets cut between the brain and the muscle is going to degenerate. That's what we call denervation atrophy. Like first, the number of the myofibrils decrease. You guys gonna read this at home, I don't wanna, but the point is, how does this work, guys? The truth is that even as you are sitting and you are not really moving too much, the brain is continuously sending a little stimulus to the muscle at all times, creating micro contractions, and that makes the muscles happy. Okay? So you need not only blood supply but nerve supply. Okay? And then you guys read this atrophy, hypertrophy, atrophy, blah 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 at home. Now we are going to the cell and the tissue level. So this is what I wanted to again emphasize for you guys, that I prefer you're gonna hear all the time fiber, 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 you know, and then suddenly you don't know which sausage I'm talking about, because they're all kind of like a fiber. Okay, so I do recommend that you stick to the name, say cell. Just skeletal muscle cell. This long, elongated, could be 1 meter long cell. Very different from other cells, right? So, because the name fiber, again, overused. Just like I told you a couple of examples, the nucleus and other examples, membrane, nucleus membrane. Because collagen fiber refers to a protein. When you say nerve fiber, which is axon, they are really part of a cell, piece of the neuron. When you say muscle fiber, you are talking about an entire cell. So the fiber has totally different meanings. Okay, so here comes the what level? The cell and the tissue level. Okay, so one thing is very important that you understand. Skeletal muscle cell is a morphological syncytium. Okay, syn is easy, means together, because that's where the word symphony is coming. So phonic is music hearing, together. Syncytium is actually the cell, the cells together. So what happened? This 1 meter long, how did this 1 meter long cell develop, guys? Large cell made by fusion of many cells during embryology. So there is a fusion of teeny tiny cells. Okay, so let's look at a little bit of embryology. There's too many transcription factors here, but I just mentioned one, the MyoD protein, that at one point, and it's much more complicated, you don't want to know. That's what I'm looking for. It stops the mitosis. So let's look at the events. So we start with mesenchyme. Did you guys learn about somates yet from Dr. Paul? Yeah, there was no embryo, so it's coming. Then from here, myogenic cells develop. Then myoblasts develop. And the myoblasts are the ones that are going to start making these little tubes that eventually becomes the 1 meter long muscle cell or fiber. So basically, muscle cells that we have right now, you can call it post-mitotic cells. What does it mean? They cannot do mitosis. The only way they can do a little bit of repair, or if you go to the gym and you do bodybuilding, that there is a satellite cell sitting there that can add more myofibril into the cytoplasm. Okay? So here's the image for you. Look at day 15. That's pretty early, right? The mitosis already stops. Here's the myoblast, multiplies, lining up, gap junctions form, then the plasma membranes all disappeared, and now you have a tube, myotube, and here comes the multinucleated contractile cell. So now not just long, but we still have the nuclei of every single myoblast. So that's why when you look at we already did that. When you look at the skeletal muscle cell, that's why it's not only long, but it's also multinucleated. The left behind from the original little cells. Okay, so this is talking to you, where is the myosatellite stem cell that can do a little micro repairs? They are actually sitting extracellularly on the top of the sarcoplasm, under the endomysium. Okay? And that is, anyway, so this is the steps, you see, repair of the satellite cell. Enter the skeletal muscle, probably the plasma membrane fuses and go in, start multiplying, the myoblast becomes, the satellite cell becomes myoblast, multiply, kind of repeat what happened in embryology. And making myotubes, and then form the fibers, again, it would be better to say myofibril, but not right now. Okay, so this is a repeat from previous lecture that you, okay, one second. Good. All right, so we repeat. You remember that we did this in light microscopy before with the mitochondria? That the skeletal, every skeletal muscle actually is a combination of 3 different types of skeletal muscle cells. I want to remind you, this is one cell in cross-section, okay? And some of them has a lot of mitochondria, some of them has little mitochondria, and one of them is between. And I just put it here for you that you understand that the lots of mitochondria is called type 1 cells. The little mitochondria are called type 2b cells, and the between is the type 2a cells. The first one can do aerobic activities. So basically when you are doing marathonic running, these cells are working. Then if you are doing weightlifting, these are anaerobic fibers, and this is what I hope physiology people are gonna teach you what it really means, this, okay? But I just wanted to touch on it. And so this is what those fibers that are working during weightlifting, and the 2NA is between. So it can do a little running, little short running, not marathon. Okay? So now here is the skeletal muscle. I think we covered everything. Multinucleation. And not only that, look at the many nuclei that push to the side. Why? Because who was making the striations? You remember? The myofibrils. And who makes the striations? The myofibrils. So all of these pink things are all myofibrils, 1,000 parallel sausage next to each other. Okay? So, and again, you remember the most interesting structures usually see in longitudinal view, just like the node of Ranvier. Here, the striation in longitudinal, not in the cross section. Now, this is the way the transverse section looks like. This is one cell guy. I put it there so you don't gonna get lost. So what is the red dots everywhere? The myofibrils, the organelles, the 100 to 1,000 inside. You can see multinucleation only on this guy, unfortunately, but you can see that the nucleus is pushed to the side. So tissue is coming up. I wanted to make so right now in theory I was talking only about cell. Now I'm coming to the tissue. How do I make a tissue out of this long elongated cells? All you have to do is put them parallel next to each other. That's why I put here for you the model. And you're going to be able to see this in the dissecting room too, that you can see the individual muscle cells. Okay? So they are parallel, just like on the macro, also in the micro. Okay, so it's repeat, guys. So it's nothing new here. You guys read it. We already covered it. The only thing I wanted to show you was that this is what I call the butcher cut. Because when, you know, let's face it, skeletal muscle is meat. Okay? So when you go to the Tamimi market, this is the most of the time you're going to see it in a butcher cut. I recommend you to check it out. Okay? And so this is one cell, okay? Nuclei, multinucleation, push to the side, and the shape of the cell is polygonal, and they are really big, 100 micron diameter. Okay? Everything is the same, we covered. Now this is, I just put here for you fun, that the same names that you're gonna memorize, you know, for OSPI at the end of MSK, has their own names if you learn how to cook. What we call culinary art. They have their own names. The most famous one, guys, is the tenderloin. You guys have, I'm sure you had tenderloin before, right? That's the best steak ever. Where is it coming from? Guys, from the psoas major. What am I doing? That's the minor. Psoas major. Okay, and here it is. And look, as the person knows how to cut it up, that's the way they sell it. You heard that filet mignon before, right? Now that's where the filet mignon is coming from, guys. Each one of the parts has its own culinary name. Okay, one more thing. When you look at the muscle, or meat, right, in the Tamimi market, you're going to see two white structures, right? One of them is going to be connective tissue type 1 collagen, right? The epineurium, epiperiendoneurium is all there. And guys, the marble is the fat that makes the meat tasty. It's all there together. Okay, now myofibrils are coming up, right? So we are at the sausage level number 2, made out of sarcomeres, okay? Now this is what I'm talking about, that don't get lost, okay? Sausage 1 muscle, sausage 2 fascicle, sausage 3 cell, 4 myofibril. So where are we right now? In the myofibril. What is it? Intracellular organelle, okay? And we already covered all this. They are filling up the entire cytoplasma, and they are the chain of sarcomeres. We covered everything here already. Now, the only reason I put this image here for you guys, because, why did I do that? I don't know. Because here you see only one myofibril. A piece of myofibril. Here you can see how 9 myofibril completely fills up the cytoplasm. I think that was my purpose. But it's not 9. You remember? 100 to 1,000. And as they are lining up with each other parallel, that's the way you're going to be able to see the dark lines, dark bands, light bands, and alternating, and you can see striation. Okay? This is the same thing. Sarcomeres are coming up. So we are here, Daniel sign is here. Sausage number 5, sarcomere, structural, functional unit of the muscle cell. What's in here? Myofilaments. Which one? Thin or actin, thick or myosin. And again, everything is protein. So here, all over again. So now, we are focusing the filaments, right? In the center, this dark fellow is the thick filament or myosin filament and represents the A band. Then outside here, it doesn't show very, now here we go, the blue guys are the thin filament or actin filaments, okay? And they're gonna be representing the light band. Just that you understand why the myosin is dark and the actin is light. Which structure would be darker? A thick structure or a thin structure? Yeah, so that's why, okay? So here is all labeled up for you. So again, electron microscopic image. Z disc to Z disc is the sarcomere, okay? And the Z disc cuts the I band in half in a relaxed muscle. Physiology people will teach you. So this is your thin filament or actin filament. Then the dark band here is the A band or myosin. And in the center you have an H zone and a M line, dark line. Okay, now this is a very good image to see the striations in light microscope and electron microscope. On the light microscope, the dark light, look, the A band, I band, pink, white, pink, white, pink, white, pink, white. Now you translate this to electron microscopy, black, white, black, white, black, white, black, white. And the white is cut in half. Now what am I saying? The white band is cut in half in the light microscope, and the white line is also cut in half in electron microscope. So that's going to be your Z line. And again, Z line is a bunch of proteins you don't want to know. But that's exactly what's on this image. And I'm going to let the physiology people, I just want to put it here for you, other than actin and myosin, these all are different kind of proteins that are maintaining the precise alignment of the thin and the thick filaments. The only famous one here, I'm sure they will cover for you, the dystrophin. If there is a mutation in this gene, that's where Duchenne muscular dystrophy happens in boys. But I don't want to talk about it much. And I don't even want to put the function there because I think they will cover it for you. So I just thought I'd give you a little, okay? Myofilaments is coming up, so we are at the end of the line, okay? So, so this is your filament. This is the thick filament, the pink, magnified here. The blue is the thin filament, magnified here. So this is one thick filament. It's made out of 500 myosin protein molecule. Okay, so this is not one molecule. One little, looks like a, what is that? A golf ball, right? Not the ball, the golf, whatever. Okay, golf bat. That's one cell, okay, one protein. And you are making a filament out of that. Okay, now comes the thin filament. Or the actin filament. You guys remember that we were not able to start on time? So I have to go on, I'm sorry, okay? Okay, so the thin filament or actin filament is basically the blue fellow, okay? The thin filament is also called F-actin, and you see how it's put together from little blue globes. That's called blue globular actin or G-actin. And the way it's like, looks almost like a necklace, right? The way it's organized, it's put into double helix, okay? So now comes it's not that difficult, but when you cut transverse section of the sarcomere, and when you cut across where the thick and thin filament overlaps with each other, that's when you're gonna see hexagonal arrangement. What does it mean? Six thin filament surrounds one thick filament, or 6 actin filament surrounds 1 myosin filament. This is when I say there is a God. That image tells me that, because you see the total beautiful organization. There's no chaos here. It's perfectly organized. Now, if you look at from the thin filament point of view, you see a triangle. Okay? So, and again, this is what we just looked at. The actin and myosin overlapping. Now, this is the M line, where you're going to see a lot of myosin plus accessory proteins. This one is the H zone. It's only myosin. And when you look at the I band, when there is no overlap between the thin and thick filament, all you're going to see is actin. Once you get used to it, it's not that difficult. So now, guys, now comes the actual molecules. That was not even on the image. So we're going to have the myosin molecule, the actin molecule, the tropomyosin, and the troponin. Okay, so this is the way one myosin molecule looks like, guys. So it has a tail, and it has 2 heads that physiology people are going to call cross bridge. And it both cross bridges has actin binding site and myosin ATP site. What does it mean ATP as? And an enzyme where it's going to cut the ATP in half to release the energy so that we can move. So therefore, the chemical energy, what is sitting in the ATP becomes what kind of? Kinetic energy, kinetic. So, what else works like this, guys? Your car. The benzene is your chemical energy. You burn up the benzene and it becomes moving the wheels, a kinetic energy. Okay, now that we got this straight, you see here is your one molecule and making the filament. Okay, now we already covered this, so I'm not going to bother with this. Now we already did this too, right? A lot of G-actin becomes the F-actin double helix. Okay, now the new proteins are, guys, what is still here is the tropomyosin, these little fellows that are a alpha helix. You see that it's like 2 chains are in a helical form. And look what the tropomyosin is doing. The tropomyosin is actually covers up these black dots. What are the black dots? The binding site of the actin to a myosin. So actin and myosin cannot rendezvous. They cannot meet. So tropomyosin is a bad guy. He doesn't let actin-myosin love story prevail. They want to meet so badly. Just kidding. Okay, now that we got this change, troponin complex is coming up. That's little. This is the only guy, guys, so far that is not a sausage. Everything is elongated and elongated and elongated. The troponin complex actually has 3 subunits. The T binds the tropomyosin, one of the 3 components. The C part binds calcium. And the troponin I, which refers to inhibition, binds to actin. This is really the bad guy. It doesn't let the actin and the myosin. Okay? Okay, so now everything put together. I don't think we need to analyze that. Yeah, this is just showing you that this is when relaxation occurs. The tropomyosin totally covers up the black dots. That is the binding site to actin. And when the contraction occurs, you see the black dots are all exposed. So now love story begins. Okay? So here we go. This is summary. Okay, now we have to talk about the T tubules. I'm sorry guys, there's a few more slides. So, where are we right now? Intracellularly, and these structures are visible only with electron microscopy. Okay, so that's why I made this here separate section here for you. Do you see the yellow guy in the middle? That's your T tubule and transfer tubule. What is this guy doing all day long? Do you see? It doesn't really show clearly, maybe here. This is the plasma membrane. The plasma membrane actually extends into the cytoplasma, bringing the action potential in to every single myofibril. And more precisely, to this blue left and right. What is the blue left and right? They are the sarcoplasmic reticulum, where calcium is stored. So this is what I usually say. T tubule wakes up the terminal cisternae of the sarcoplasmic reticulum. This is called terminal cistern. Okay. Now, and this is what's going to blow you away, but I promise you that physiology people are going to do it. So, what kind of proteins are sitting in the T tubule? What kind of proteins in the, because it's a plasma membrane, remember that, it's a plasma membrane. What kind of protein is sitting there? Initially, it has to be voltage-gated sodium and potassium channels because its action potential is coming down. But when you're coming closer to the SR, or terminal cisternae, then a new protein shows up. And this is called voltage-gated or sensing L-type calcium channel protein. They also call it dihydropyridine receptors. Isn't that an awful name? So that's when you say, okay, excuse me, is this a channel protein or a receptor protein? And the answer is, it's like the shampoo, 2 in 1. Okay? So it's, if you have to imagine a protein, you don't want to know how many subunits and all of that stuff. Just one time Google it and you're gonna, okay, anyway. The point is, so if the molecule binds to the receptor, blah, 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 that's when conformational change occurs and channel protein opens and calcium goes. That's what I call the 2 in 1. Okay? Where are these T tubules are located? At the junction of the A and I band. Why is this? It's not really not that important, but it's just really weird that in cardiac muscle, everything is the same except this. The T tubules are going to be at the Z line. I don't know why. Okay, next is coming up. Now we're going to focus on the sarcoplasmic reticulum, which is, we learned that before, is a modified smooth endoplasmic reticulum. You see, here's the zoom sign, and I magnified here, the same thing. So here's your triad again. I'm sorry I didn't say triad yet, but the one T tubule and the left and right sarcoplasmic reticulum together, the 3 is called triad. Okay? Okay, so now that we are done in here, okay, so T tubule already woke up the SR, okay? So what's gonna happen now? We have some new proteins because now the calcium has to come out from the SR. So here comes another shampoo, okay? This is called ryanodine receptors, or RYR, and they are calcium channels. So 2 in 1. So what does it mean? So when the T-tubule wakes up and whatever molecule physiology people tell you every step, binds to the receptor of this protein, the calcium channel opens up, calcium exits from the SR, so now the muscle can contract. The point is here, and they're gonna talk about it, I promise you. So I just wanted to head on for you. So where are these proteins? Right here, where the two guys are looking at each other. Okay? So the T-tubule wakes up the SR. Okay, so this is just a summary of the same thing, that of all the proteins that I covered already. The voltage-gated sodium channels, the DHP receptor in the T-tubule, the rhianodine receptor that is also, and both of them are calcium channels. Do you see that? Receptors and channels. Then inside SR, there is another protein called sequestrin that binds calcium. Okay, holds it there. And finally, the SERCA here. This is the called sequestrin, I put a CAL there for you. And the SERCA. The SERCA is going to be that protein that is not at the junction of the T-tubule. Further down, where the contraction of the muscle is finished, the calcium is going to go back to the SR. That's what SERCA does. Again, sarcoendoplazmic calcium antiphysiol, SR calcium antiphysiol. So, okay, so all of these things are coming in physiology. And mitochondria between the myofibrils, this is myofibril 1, 2, 3, 4. You can see the Z lines and beautifully colored, the same thing. Now, I wanted to touch on one thing, that when you go into sarcoplasm of a skeletal muscle, as I told you, very little sarcoplasma is left because the myofibrils are filling it up. But there are going to be 3 structures that are located there as a storage for the instant. When you start moving, you need to have instantly oxygen and instantly glycogen and instantly energy for the movement, especially when you are doing, let's say, a marathon run. So the first couple of seconds or minutes, only God knows, has to come from inside the cell. Eventually, the oxygen is keep coming from your breathing. As you are running, the breathing activity is going to start matching your exercise, but not immediately. So the instant oxygen is going to come from this hemoglobin-like protein dissolved in the sarcoplasma and binds oxygen, stores oxygen, so that when you start moving, it's ready to go. This is what I call instant oxygen. You see? So there are 3 instant I listed for you. The myoglobin, creatine phosphate, you have to learn that in biochemistry. This is instant energy. This guy gives the phosphate to ADP and makes ATP right on the spot. Okay, and then of course glycogen. We talked about glycogen storage in muscles and liver. Immediately breaks down to glucose, and the glucose becomes eventually ATP. Now, let's talk about, I think this is the last section, so I appreciate your patience. The innervation of skeletal muscle. I already told you that they cannot live without motor input, right? So that's what I'm showing you to you. The motor innervation of skeletal muscles are called the somatic nervous system. Okay? And what I'm showing you to you where the neuron and the muscle meet, what you're gonna call neuromuscular junction. Okay? So what do you see here? 1, 2, 3, 4, 5, 6 skeletal muscle cells. You can see the striations. So they are not myofibrils. This is where you have to kind of be careful, right? Okay, and this is the more motor nerve comes in, start branchy, final branching, and these are, guys, the synaptic knobs. You learned about synaptic terminal from Dr. Aftab, right? So here's your synaptic knob on the light microscopic image. Here is on a drawing. So this is your end of the motor neuron. Now comes the muscle. I didn't even cover this part because it's just getting too much. I leave it to the physiology people. But I wanted to remind you that what you cannot see on the light microscope is the T tubules and the SR. Because the moment the action potential came across from the nerve terminal and entered the muscle, where is it going to go first? To the T tubule, right? And the T tubule brings down the action potential and wakes up the SR left and right in the triad. Okay, sensory innervation, guys. And again, I can talk probably another 20 minutes on both of them, and I said, forget it, it's just getting too much. But there are stretch receptors, guys, in your muscles, okay? One of them is called muscle spindle, which is right in the middle of the, so to speak, the flesh of the muscle, buried inside and sensing the stretch. What is the whole idea? If a stretch is too much, the brain is gonna stop it so you don't tear your muscle in half. So there's gonna be a reflex, which I put it here for you, reflex, that is basically, you're gonna drop the weight in the gym because you can't handle it. It's basically a reflex from your brain. And there's another stretch receptor that is buried in the tendon, doing the same thing. So here's your nerve ending of the sensory neuron. So where is this sensory neuron is going, guys? Where is this little fiber is going? Are we doing special sensation or general sensation? So this guy is heading to the dorsal root ganglion, okay? And from there, blah, 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 blah. Same thing here. Here is more obvious, you know, the structure. Here, let's call this is the nerve that is going to the dorsal root ganglion. Okay, repeat. We did this before. This is your heavily myelinated axons in the somatic nervous system, somatic motor and somatic sensory. To speed up, we need fast action potential for both, for sensation and both for movement. And you guys remember? Say hello. This was in this morning on the exam, right? Okay, compare the three muscles. This is what we... Okay, this is what we're gonna do in the lab, okay? All right? And this is the last two slides, what I already mentioned to you guys, that for some reason, cardiac muscle, slightly different. Instead of triad, you're gonna have dyad, meaning T tubule and one SR. The skeletal muscle was T tubule in the center and two SR left and right. And you remember the other one I told you, they're gonna be, the T tubules are gonna be at the Z disc, not at the AI band. And guess what? I put it a table for you in case, you know, the physiology people decide to use it. And it's